Thanks, Jeff. So Peter approached me and said, hey, how about doing a forum? And I thought, okay, well, the last forum I gave you was on genetic engineering. And many of you, I think, know that in my day job, I'm not a photographer. I'm actually a professor of molecular biology at the University of Southern California. And so I thought, well, what other sorts of topics could be kind of fun from my professional life? And my wife, Lisa, suggested epigenetics. Cats and rats, why Penny's cat is a tortoiseshell, and why licking rats makes a difference, and what does it mean to us? And this is a topic that pops into the news now and again, but isn't really very well explained. So I thought I'd give you a very simple explanation for this wondrous new example of complexity and how our biological systems work, and how this has relevance for us. Even if you're not a scientist, it has great relevance to us. So I'm going to go ahead and share my screen. And that's not the right screen. Stop share. Desktop 1, desktop 2. There we go. That's the one. Share that one. And can you guys see that correctly? Title slide? Okay. Epigenetics, the mystery on top of our DNA. So epigenetics, epi means on top of, and this is a relatively new field in understanding how our genes work, dating back about probably about 25 or 30 years to really have exploded. And this is kind of a little inside humor here. This cartoon says, if they ask you anything you don't know, just say it's due to epigenetics. And little science humor there. But so, as we understand things, when we have a simple model for how we think the world works, inevitably it's wrong, because the world is far more beautifully complex than a simple model can capture. And I hope that I'll convince you that this is kind of a fun thing to think about, and adds to the beautiful complexity of our world. But just to remind you of some basic biology. We, as humans, have 2 copies of all of our genes. We got 1 copy from mom and 1 copy from dad. So the gametes, the eggs and the sperm, each carried a single copy, and these fuse to create an embryo, which undergoes division and specialization. And when we start out with just these 2 copies of each of our genes, we have the genes are packaged onto chromosomes. We have about, current estimates, around 25,000 genes make up a human. And as an embryo grows and develops, some genes in a particular cell type are necessary. Your liver cell needs to express liver enzymes, but your liver cell doesn't need to be expressing genes involved in brain function. Yet all the genes are present in all the cells. Some of them need to be turned on, some of them aren't going to be used. So we know that the cells of our body, what we call somatic cells, have these specialized functions. And of course, we also have the germline cells, where single copies are packaged to be delivered to the next generation. Now, many of you may know that the history of genetics was written in the late 19th century by a monk named Gregor Mendel in a monastery in Brno. And he crossed peas together to try to understand the basis of heredity. And he came up with some basic understanding, which is absolutely true. And that's that genes aren't blended but discrete. We get a copy from mom and a copy from dad, and they don't somehow blend their information. They each are providing us distinct information. We get 1 copy from each parent. And sometimes we have what we call recessive traits, which are covered up by dominant ones, but we'll see in the next generation. And what do I mean from that? The simplest example that we often use for this is oversimplified, but it's eye color, okay? And I think many science classes in school teach eye color as a basis of genetics. Brown eyes are the most common in the world. Blue eyes are what we would call recessive. Blue eyes actually arose about 7,000 years ago in a population in Northern Europe. And there's some theory about whether it gave people in northern climes some advantage to have lighter colored eyes. But we still see today that lighter colored eyes tend to be found in populations based in Northern Europe. Now, blue eyes are, of course, quite frequent especially in white populations of Northern European descent, but brown eyes are as well. Now, my dad has had brown eyes, but I have blue eyes. Well, actually gray, but technically blue. And how did that happen? It happened because dad had one copy of a gene for brown eyes and one copy for a gene for blue eyes. His mom actually had blue eyes. So one of those copies came to me. Which one? Completely random. I happened to get dad's blue eyes gene. My mom has blue eyes, so I ended up with two blue genes and thus blue eyes. My oldest brother has brown eyes. 50-50 chance he got dad's brown-eyed copy, so his eyes are brown. So the blue gene here is present. It's there in dad, but we don't see it. It's like it's hidden. And so we call that a relationship of dominance and recessiveness. Now, we can often rely on having a good copy if there's a bad copy of a gene. We have some random number of genes that don't work, but most of them are covered by the fact that we have an existing good copy. So just having a good copy present may be sufficient in many cases if there's one that's missing. Talking about genes, Mendel had no idea about DNA. It wasn't really until the 1940s we knew DNA was what genes are made out of. And so the basic, what we call the central dogma of molecular biology is based on understanding that genes are encoded by this molecule DNA. And DNA is essentially a library. It's information. It's how to build something. The thing you're going to end up building are proteins. And that's the things that make up our skin and all our cellular functions and the enzymes in our liver and so on. Those are all proteins. And the DNA is the library of directions of how to make any protein necessary. The DNA is copied into an intervening molecule called RNA, which is then translated into the protein. So the DNA is this library that exists in all of our cells, and all of our cells have all the DNA. We know a lot about this central dogma of molecular biology, including exceptions to it. But we understand that in some cases, RNA could carry genetic information, such as in, COVID virus is actually based on RNA, not DNA. But for the most part, DNA contains we think of containing a library. People have been studying how genes get turned on and turned off, and we thought we understood it pretty well. And then about 30 years ago, we realized that there was a little more complication to the matter. We got in close, and the simple model turned out not to be quite true. And the complication comes because DNA, there's a lot of DNA in our cells. If you stretched all the DNA in our cells out, it would be about 2 meters long in each cell. So you got to package it so that you can get it into those cells. And we know that the DNA gets packaged by wrapping it around proteins in order to compress it and consolidate it into these structures that we call chromosomes. So we have a DNA, which is a genetic sequence of letters that's packaged onto these proteins. Thought we understood that. In the 1960s, people discovered that sometimes there were chemical modifications to the DNA or to the proteins, and they thought maybe these just happened, they're not really important. But now we realize these chemical modifications are actually really important. Some of these little chemical modifications happen on the proteins that the DNA is packaged with, and some of them actually happen on the DNA itself. And what these chemical modifications do is provide information on top of on top of the DNA sequence that modifies how it's accessed. Because once the DNA is packaged, we need to access it all the time. So this access can affect whether or not a gene can be turned on or off. You package it up in a particular way, the gene doesn't get turned on at all. And so starting about 30 years ago, we realized that these chemical modifications actually had profound effects on how our genes get used. Which books in the library are available for you to pull out and read? These modifications may turn on individual genes, they may affect one copy, but they actually turn out to be essential as our cells make all these different functions. So our liver cells don't express genes involved in, or don't read the books required for nerves, because those books are packaged away by these chemical modifications. Brain cells don't express liver enzymes, and those liver enzyme genes are all packaged away by these chemical modifications. So we realized that as a community of scientists, that this access was affecting whether genes could be used or not. The genes are still there, Mendel was right, but whether or not we see them turned on or off depends on these little chemical changes. Now, what makes students who've learned the central dogma of molecular biology particularly uncomfortable is there's a certain randomness to this. Sometimes things can get turned on or off by accident. And we call this variegation. This is a picture of a Drosophila fruit fly. You've probably seen these guys flying around in the summertime in your kitchen, and typically they have a red eye. And it was discovered a particular version of a fly where some of the eye segments were red and some of them were white because the red pigment wasn't being expressed. All these cells are identical. They have all exactly the same genetic information, but randomly, some of them had switched off the red producing pigment. And so this makes people very uncomfortable because it means that these changes can occur over time with a certain degree of randomness. Now, if we just think about these little chemical modifications that change over time, they can change the functions of our genes just randomly over time by chance. People get very uncomfortable about that. But what it means is that the pattern of modifications that we have on our genes in our different cells differs. So, for example, identical twins aren't identical at the level of their epigenome. So the patterns of some of the genes that are expressed will actually be different. Now, this has relevance to human health because sometimes some of the genes that get randomly switched off may be genes that are important for other reasons. So we know, for example, that there are epigenetic changes associated with some forms of cancer. These epigenetic changes may be modifiers of cancer and in some cases actually drivers of cancer. These may be affecting gene expression, that is, which books are being read, and also the function of chromosomes, although I haven't really talked about that. So epigenetics turns out to be quite important, and it turns out that some forms of chemotherapy for particular cancers target the enzymes, the proteins that make these epigenetic changes. So let's think about some real life examples that are kind of fun, not just Drosophila fruit fly eyes. Let's think about people and cats. One of the big differences in the genomes between women and men is that women have 2 copies of the X chromosome and men only have 1. So men have an X and a Y chromosome. A Y is a very small chromosome, and women have 2 copies of the X. So we want to balance the gene expression from genes on the X because we don't want to have too much or too little, right? So how do you actually normalize this? Now, men become vulnerable because they only have 1 copy of the X. So if any of the genes on their X are bad, they don't have a backup. And just as an example of what happens with this, hemophilia and red-green color blindness are what we call X-linked traits. This is an example of the royal houses of Europe where hemophilia became a problem. Hemophilia, this particular form of hemophilia, is on the X chromosome. This is Queen Victoria up here. She carried a bad copy of an X chromosome, but she also had a good copy, so she didn't have hemophilia. Yet men in the line who inherited the bad copy of her chromosome, indicated by this dark red, would be at risk for would have hemophilia. And here's the little prince of Russia, the young son of the Tsar, who rather famously had hemophilia. So Queen Victoria managed to seed hemophilia throughout the royal houses of Europe, affecting only the men. Now, but here I just told you, though, that we have to balance the expression here of the X chromosomes. So how do we do that? And we do that in women by randomly selecting one of them to turn off using epigenetics. So if we start with a zygote or an embryo that has 2 X chromosomes and is female, at a fairly early stage in its development, randomly in different cells, one or the other of those X chromosomes will be switched off by an epigenetic mechanism, completely switched off. So that means that all the XX women are basically mosaic. That is, some of our cells are running on one of our X chromosomes, and some of our cells are running on the other X chromosome. Why this? The classic example to explain this. This slide, actually, next slide, comes from my lectures on this subject. Is orange cats. The orange gene is on the X chromosome. This is my cat Chase. He's an orange tabby. He has 1 copy of an X chromosome that has the orange gene, and he's a very handsome orange cat. Penny's cat also features in my lectures. This is Mitzi, Penny's cat. Mitzi is a tortoiseshell. A tortoiseshell, or calico if it has more white, is a mixture of both orange and black. One orange chromosome, one non-orange chromosome. So you can see Mitzi's face here is partially orange, but also partially black. And the reason for that is that early in Mitzi's development, randomly, some of her cells shut off the orange chromosome and some of the cells shut off the black chromosome. And you can see essentially this whole lineage of cells that is expressing orange and a lineage of cells that is expressing black, one copy being randomly silenced. Now, people mistakenly think that you can't get orange females, and that's not true. This is, it's possible to get an orange female if she has an orange dad and a mom who's a calico or a tortoiseshell. So 50-50 chance that she'll get 2 copies of the orange. Example of this is Chase's sister, Hershey. Hershey is a very elegant little floof, and she is an orange tabby as well. And I can tell you that Chase and Hershey had to have an orange father, and their mother was either orange or calico, tortoiseshell. So we have 2 X orange chromosomes here. The likelihood of getting a female orange cat simply depends on the likelihood of this particular mating taking place. And if you cross an orange female to an orange male, all the kittens will be orange. I swiped this picture off of the internet to show you that. So orange female cats are uncommon, but not unusual. So it just depends on how frequently we see orange chromosomes. And this is because of epigenetics, randomly shutting off one of those chromosomes. Male tortoiseshells, on the other hand, are extremely rare. And that's because the only way you can get a male tortoiseshell is if he's got 2 X chromosomes. This is a picture that as a cat that was hit the headlines in Great Britain about 5 years ago. They named him Burrito, and he is a tortoiseshell male. And he's a tortoiseshell male because he had an abnormal chromosome segregation, so he actually got 2 copies of his X chromosome. Now, the system for shutting off randomly one of those copies works just fine. If you have more than 1 X chromosome, that system is basically built into the X chromosome. So he randomly shuts off one of the other of these. This is this abnormality of 2 X's and a Y. It occurs in humans as well. It's called Klinefelter syndrome. It's surprisingly common. Individuals who have 2 X's and a Y are apparently male, but they're often infertile and they have reduced secondary sex characteristics. So this is going to be not only uncommon, but unusual, whereas an orange female simply depends on how many orange cats there are in the vicinity. So we now understand the basis of this and the fact that women have randomly shut off one of their X chromosomes. So how come Queen Victoria didn't have hemophilia? Expressing it that the clotting factor in half of your cells is enough to keep you clotting properly. So, can the environment affect epigenetics? Here's another cartoon. How do you want to play this? Nature, nurture, or a little bit of both? Well, the answer is it's a little bit of both. Mendel tells us how our genes are inherited, but the epigenetics may affect how they're used. And it turns out those epigenetic modifications can be susceptible to the environment. A study was done to look at rats. Now, rats, if a rat mother is calm and relaxed, she spends a lot of time grooming her pups. If she's a nervous mother and she's upset and anxious, she may not groom the pups. So the pups that are groomed and get licked have different behaviors than the pups who don't. So investigators looking at this to see if there was a difference in how genes were expressed in pups that were from happy mom versus not so happy mom, went ahead and looked at these chemical modifications on genes that are involved in behavior. And it turns out that happy mom has low stress, her pups have low stress, and if you look at the pups' brains, they are expressing genes that are calming. The unhappy mom, high stress mom, her pups were also anxious, and it turns out that there were chemical modifications to their genes that turned off genes that were calming, and so now they have increased stress. So here's a particular gene that's on low stress in the happy rats, and here's that gene is chemically modified and switched off in the high stress rats. So the next experiment was to ask, what if you switch the pups? Let's take Mendel out of the equation. Let's take the pups from unhappy mom and give them to the easy mom and vice versa and see what happens. Do these epigenetic changes track with the behavior of the mom? And the answer is they do. So these pups who came from the unhappy mom were mellow. They had the pattern of epigenetics that are seen patterned by the mom here. These pups that came from the happy mom and were given to the anxious mom turned out to have the chemical modifications occur based on her behavior that changed their gene expression. Once they have those chemical modifications, they tend to stay there. And those pups themselves tended to raise pups that had anxiety. Can you fix it? Well, these are rats in a laboratory, so they could treat them with a compound that would take these chemical modifications away, and asked what happens if you take those chemical modifications away. Can you de-stress those pups by removing the chemical modifications? And the answer is yes, you can. So having identified this particular set of modifications, we could pattern those modifications and see that they occurred in response to the behavior or the environment of the pup was growing up in. Now, does this have relevance to people? Yes, it does. Simply holding on to an infant or not can lead to these same kinds of changes. So an attentive human mom who's nurturing her child is similar to the rat mom who's licking her pup. Whereas a nervous mom who's not interacting with a child who's not undergoing that proper nurturing will end up being high stress. A number of years ago, you may recall, they discovered, before the fall of the Iron Curtain, they discovered that there were orphanages in Romania where children were essentially growing up completely deprived of proper nurturing. Certainly, those kids had these sorts of modifications occurring that were affecting how their genes were used. More examples. Child abuse leads epigenetic changes on the brain. And in looking at suicide victims who were abused, it was determined in autopsy that they had specific modifications on genes that are involved in some of these responses. And in this case, some of the same genes that were seen in the rats. Psychiatric patients may have chemical differences, modifications that affect gene usage that may impact this. So one of the questions about this is, well, I don't know if you're aware of this, but at a point of time when people were starting to study genetics, a Russian scientist named Lamarck tried to argue that genes could be affected by the environment themselves. And so he said, giraffes who reach higher will end up having acquired traits that get passed on to their offspring. Well, we know that's not true. It's not affecting the transmission of the genetic information, but it may be affecting, the environment may be affecting how that information is used. Can these modifications be transmitted directly to the offspring? For the most part, no, because when eggs and sperm are made, all these modifications get scrubbed for the most part. So we're not transmitting these on to the next generation of organisms, but the behavior and the environment around offspring can have this effect. I don't know if you remember a few years ago, there was a big thing about BPA water bottles. We weren't supposed to use BPA water bottles, and we all threw away our Nalgene water bottles. BPA turns out to affect this kind of chemical modification, and it leads to a change in DNA modifications, epigenetic changes and modifications that were part of the reason that BPA was considered harmful. So our environment affects these signals on top of our genes that affects how we function. So epigenetics is information on top of the genetic information that affects how it's used. Going back to the analogy of genes being a library, epigenetic modifications may shut some of those books away into a locked cabinet. Epigenetics is part of our normal programming. We see it with differentiation as our cells specialize as we develop. We see it in the X chromosome inactivation of mammalian females. But there's also the potential for a random component. It can drift and change with age that can end up affecting gene expression and gene usage as we age. It can be influenced by the environment, including our diet and our behavior. And each person's epigenome, that is the pattern of modifications on top of their epigenome, is unique. So cats, rats, and Mendel is what we go with this. There's a couple of accessible articles about epigenetics if it's something that interests you. There was a great article in the New Yorker in 2016. So if you just search epigenetics in New Yorker, you can come up with this. It was a nicely written article by Siddhartha Mukherjee, who's a physician, but it's a really good article. And then there's a nice website at Sloan Kettering, What is Epigenetics and Why is Everybody Talking About It, which also goes into some of this in more detail, if it's of interest to you. So we are wondrously complicated, and every time we think we understand how we work, we discover that we are only just scraping the surface of this. So hug your children, don't drink out of BPA water bottles, and I will be happy to answer any questions that you may have.
You might look through the chat, Susan.
Okay.
There were some little conversations. Thank you. This is absolutely fascinating.
Okay, I'm going to look at some of the questions. So there was a question from Jeff. Why don't women have a single problem without a copy of Y? Well, it turns out there aren't any essential genes on Y for life. The Y chromosome is pretty minimally carrying things that confer maleness, but those are not essential for life. So yeah, women have no Y at all. That's absolutely true. And there are examples of kind of strange rearrangements of chromosomes in which pieces of the Y chromosome may be accidentally fused on other chromosomes, which now confers maleness even in the absence of a Y. So chromosomes, more of this wonderful variation. Okay. Oh, yeah, there's not much on the Y. Okay. Okay, I see people. What if the person is XXY? Yes, Klinefelters. Most of our chromosomes, we really can't tolerate abnormal numbers, or what we call aneuploidy. There are rare examples of where having an abnormal number of chromosomes is compatible with life. Down syndrome, for example, which has a profound, we think of having a profound effect, is actually 3 copies of chromosome 21. It's actually one of the mildest examples of that, because nearly every other chromosome, if you have 3 copies, you're dead. The exception is the X chromosome. So we see these rearrangements and strange copy number variations in the X chromosome. So there are individuals who have just a single X and no Y, and they have, I mean, they're viable. I mean, so but sex characteristics are kind of superficially female, but no further development in secondary, and it's often associated with some other phenotypes as well. As I said, XXY, Klinefelter syndrome. XYY, there are examples of multiple copies of the Y chromosome. There was a discredited study a number of years ago that argued that men who had multiple copies of the Y were more aggressive and overrepresented in prison populations, but that turned out not to be true. And extra copies of the Y don't particularly seem to be a problem. But it does not undergo silencing the way extra X's do.
I wanted to add a comment if I could.
Yeah, sure.
Underscoring your points, there's work by, for example, if a mother during pregnancy is stressed, it triggers through epigenetics a homeostatic mechanism that Bruce McEwen from Rockefeller had delineated, such that the fetus is better able to respond, adapt to stress once the mother is delivered. So a little bit of stress can be adaptive through a
yeah, and so there was an example of so there is definitely a component of epigenetics affecting in utero. At the end of World War II, there was a period in the Netherlands where there was food shortages, and they called it the hunger winter. And they found that women who were pregnant during that time, there were sequelae to their offspring. And if their offspring were daughters, there were also sequelae to the grandchildren. And the reason for that is that eggs, the eggs that we carry, women are born with all the eggs they're ever going to have. Unlike sperm, which are continually made in the male system, women have all the eggs they'll ever have upon birth. So when those women were starving in the Netherlands and they were pregnant, that was influencing their fetus. And if the fetus was a female, it was potentially also influencing that fetus's eggs. But there's no evidence for epigenetics going beyond that kind of level. I see a question, males are highly represented in many birth defects and disabilities. Any thought? You know, aside from certain examples of the X chromosome, which actually has some substantial, just to kind of make this a circular argument, there are components of the epigenetic system on the X chromosome. So some of those X genes are genes that are involved in reading these modifications. And so there's an issue, many of these actually end up having issues in men, particularly if you have a defective copy, there's no backup. So some of those are actually affecting epigenetics more broadly. So there may be something there. I see, okay, Penny, a calico cat, if the expression of orange and black is random, why does she have some sharp delineation between colors? Yeah, okay, so what you're seeing there, I would love to have one of my cats to describe it, but the pigment that the orange and the black pigment in the cells, when we start and are developing as an embryo, the cells that are going to form our skin and the fur on our cats are going to start at this. Oh, I have a cat. Thank you. My loveliest, charming assistant has provided me with a cat. This is Hershey. And you can see Hershey's got quite a bit of white on her. And if you have cats with white on them, you'll see they are white generally on their bottom side. You tend not to have a cat that's white on the back and colored on the bottom. And the reason for that is that the cells that are carrying the pigment start out on the back and start migrating down away from the spine during development. The white is a gene that inhibits that migration, so that's why she's white on the bottom. But just as Penny noticed with the sharp boundaries on Mitzi, you can see very sharp boundaries here on Hershey. So some of these cells, the pigment producing cells, have this very sharp boundary here. So what the difference is, thank you, Hershey. The difference is that the sharp delineation is because you have a group of cells that are derived from an orange expressing, and they kind of all travel together. And then right adjacent to them, you have a group of cells that derive from a black expressing. And so there's a boundary because they're in a cluster. Mitzi has enough white that those boundaries are really quite sharp, and she's got large spots. Some tortoise shells are much more diffuse. They don't have any white at all, so the cells intermingle much more. We see lack of methylation of factor in autism, that it is very common for people to have one or more genes for this inability to methylate various B vitamins. Well, B vitamins are actually involved in the production and use of methyl groups. And you know, people tell you that you should eat your green vegetables. That's because they're a source of folate, and folate is a source of maintaining proper methylation levels. I think the real interest here is people increasingly wanting to understand what changes in these modification patterns may be associated with differences in behavior, but it's a little hard to do that on a brain-wide scale on a living patient. So I think there's still a certain number of questions that need to be addressed about that.
I have a question, and I'm not nearly as smart as most of your other question people. So, but I got about 10% of what you said, and I'm trying to still figure out, I like the rat example, nature and nurture. I'm getting like there's some difference between epigenetics and say a chemical reaction in the brain. I got that from your presentation. Can you say more about why that's important and what applications that might have? Like, what difference does it make that this is different? That epigenetics is not a chemical reaction, and how might that make a difference in? Why should I care?
Right. Okay. So I think one reason to care is that we do have evidence that, you know, the nurturing and the environment of a child is raised in similar to the rat being groomed by its mom, leads to changes in its gene expression that are lead to low anxiety. So again, using my analogy of your genes being kind of a library, you'd like to have access to the books that are leading to a healthier, happier life. Whereas if those books are locked away by these epigenetic changes and you can't gain access to them, then you don't have that, that low anxiety situation. So what we'd like to understand, I think broadly, and of course I'm a basic scientist and I'm not a clinician, so I'll yield to our clinicians in the audience. What we'd like to understand is what some of those genes are, and are there ways that we can reverse these modifications that would allow access to that component of the library. I think another example of this, as I mentioned, is that some forms of cancer are linked to inappropriate chemical modifications. And we know that there's been evidence for using drugs that change those modifications as part of chemotherapy. But of course, this is a double-edged sword because, you know, if you have a modification on gene X that you don't want, but you have a modification on gene Y that you do want, you don't want to go in there and take all the modifications away. Because some of those you need. You don't need your brain cells expressing liver enzymes, for example. So it's very hard to think about how you can target these changes specifically to the genes that you want to affect without having a disastrous effect overall. Does that help, Jeff?
Yeah, I guess where I was getting confused was if to turn the genes off and on, all you had to do was switch the babies, then it doesn't seem like it provides much of a benefit, because if the genes just switch back on or off when you move them again.
Well, that's the thing. Okay, if you move them, if you switch them at birth, you could clearly see that was a thing. But could you change it again when you switch them back? And no, not so much. Once those changes were imprinted, they pretty much have a tendency to stay because there are mechanisms to keep them going. So it's not infinitely plastic.
Okay.
So another consideration is the there's a resiliency factor. There's a resiliency gene, the long arm of the serotonin transporter, which is passed through the mother. So if you've got the long arm serotonin transporter, you're more resilient to stress or these epigenetic modifications.
Yeah. So yeah. So this is all working on top of you know, the genes we get. So if you have if you already have a mutation in your de-stress gene, so even if that gene is turned on, it's not working properly, this isn't going to help. So Mendel is like the genes we have, it's like an artist's palette. All right. If I can't paint a picture of the desert if all I have is you know, greens and blues. So I'm limited by what the palette is. This is something on top of that palette that then is affecting the choices that get made. Can meditation or prayer affect epigenetics? That's an interesting idea, whether or not, I mean, that, because again, can our own behavior affect our own epigenetics? I think there's some interesting ideas about whether or not that's the case. I don't know. Implications for tendency to obesity and early traumas such as underfeeding. Yes, absolutely, Penny. There are implications for obesity and trauma. In fact, there are some links that have been made between distinct epigenetic changes in a parent that impact an offspring. And that BPA example, and I said that BPA had an effect on methylation. Turns out that one of the systems in which that was investigated was a particular mouse model where if you fed them BPA, they became very fat and cancer prone. So there were changes in the epigenetics that were impacting a lot of the physiology of the individual. I see Polly has put into chat a TED Talk that is childhood experiences and effects on long-term health. It's kind of relates to some of this. But I think I'll just leave you with the key idea that our environment does influence us as an organism in a myriad of ways. And we're beginning to tease apart what some of these are. And I'm fascinated to think in the next 50 years what new wrinkles we will find and how our genetic library is used and changed.
Susan, I have a question. Susan, didn't that hunger winter in the Netherlands, didn't that lead to a propensity towards diabetes?
I think so. I don't remember the details, but there's something like that. There's been examples of epigenetic modifications and responses that are associated with more cancer-prone syndromes and other conditions as well.
Is this where your research is done, Susan? And can you say something about that?
So my research is at a very fundamental level. We study the fundamentals of how cells deal with stress that affects their genome. My work on epigenetics looks at some very specific aspects of how these modifications impact the ability of cells to repair their DNA. Epigenetic modifications are also associated with the mechanics of how chromosomes are duplicated and separated between dividing cells, and some of our work investigates that.
Wow.
Yeah, I would just like to thank Susan and say you are giving such a great illustration of how science works, which is, you know, there are observations, there's patterns, you recognize patterns. Meanwhile, we have the tools that we have at the time. And say there were patterns that were recognized at one point that we didn't have the tools to investigate at the time. And we thought the central dogma of molecular biology was everything. And then, you know, then you start seeing stuff that makes you go, huh? And basic scientists like Susan then go and keep unpicking and developing new tools. And then we have ways of asking questions that we can go, huh, you know, nature, nurture. Well, we have some more scientific tools to say, well, some things do seem to be nature that seem to be an effect of the environment. Doesn't mean it's answered the question, but we just have this continually evolving tool chest that let us ask more interesting questions and find even more fascinating answers. So thank you, Susan.
Thank you, Donna. And thank you all for coming in here and letting me tell you a little bit about this. And yes, I do use pictures of the cats when I teach this subject, because I teach a course in epigenetics in the fall.
So Mitzi's a kind of Henrietta Lacks of cathood.
Yeah. You know, and it's a great example, but I think it's really quite remarkable to think that it turns out that while cats and X chromosomes make it pretty clear, there's a certain amount of this kind of epigenetic modification that makes all of us partly mosaic. And some of our cells were expressing one copy of one gene, and then other cells were expressing other copies. And there's infinite complications of what we call imprinting, which I will not bore you with. But we are, this simple model that we have all the genes in two copies, and you know we have both those copies working, is not true. We're far more complex than that. This is just so fun. Oh, Bill points in a comment, if I can just take that look a little bit more. Bill says there's a discredited theory that parents make their male children gay, and the argument is that being gay is primarily genetic, but some parents may sense their child is gay and respond in ways that affect expression of that gene. Well, it is thought that that our height, okay, why is Jeff tall? About 180 genes are involved with Jeff being tall. So something as complex as human behavior and sexuality is going to be infinite number of genes, well, not infinite, but a large number of genes affecting it. There are some arguments suggesting that that being gay is affected by the environment inside the womb. Twin studies make it clear there's a genetic component, but it's not limited to that. But it's far more complex, and the idea that there's a gay gene for any of us who are gay is definitely not the case. No geneticist would think that that there's going to be a blue eye, brown eye example for sexuality. We're far more complex than that.
Susan, no wonder that you were recently honored as a distinguished professor. You are just a fantastic teacher. Thank you. Well, it's easy to talk about things that you care about, and I really enjoy talking about my work. And I thank you, Peter, for approaching me, and thank you, my wife Lisa, who said cats and rats. Come on.
Well, as so many have said, thank you again for Susan. It's so wonderful to have you be able to do these talks for us occasionally and show off your photography as a daily part of our common life, but to show off your professional side. Well, your photography is pretty professional as well, but your professional scientific side as well, which is just amazing to hear you talk and make these things so accessible. And so we all appreciate you very much and are glad you're here.
Thank you.
Again, next week, I hope you'll join us. We'll have the CEO, Micah Parson, will be here with us from the Museum of Us. So I hope you'll join us for that. And with that, we'll see you in worship in about 45 minutes. Take care, everybody.
Thanks.